Okay, so it's 9 o'clock. Welcome everyone to our today's museum seminar. So again, I think this week is our last week for this dual seminars week over the summertime. So again, I want to thank everyone online with us during all summer, and particular during the weekend, being with us, having fun with different sciences. And also, I want to have a big thank for our today's speaker, Dr. Fu Tianmin, actually being with us during the weekend evening. So it's really my pleasure to welcome our today's speaker, Dr. Fu Tianmin from Department of Biological Chemistry and Pharmacology from Ohio State University. So Tianmin is already an old friend of our platform. He used to give a talk, I think 3 or 4 years ago, back then. But then I think he made a huge progress on his research program, which I think is really quite amazing for all of us. So Tianmin completed his undergraduate study in Shandong University, continue with his PhD training in biochemistry and molecular biology in Beijing University. So after he complete PhD study, he moved to US, study with Dr. Wu Hao on biochemistry and structural biology as a postdoc fellow. So he stayed with in Dr. Wu's lab from New York with her in Cornell, and then moved to Boston to Boston Children's Hospital, Harvard Medical School. And then from 2020, Timmy started his own independent research program as assistant professor in Department of Biological Chemistry and Pharmacology in Ohio State University. So the general long-term goal of Timmy's lab is try to understand the logic of cell signaling in inflammation, cancer biology, and then neuron degeneration. His lab's recent focus is on lysosomal signaling, which plays critical roles in immunity, cell metabolism, as well as neuron degeneration. And then they're using multidisciplinary approaches, including biochemical reconstitution, cryo, electron microscope, X-ray, structure biology, cell biology, and then pharmacology to study the assembly and regulation of lysosomal signaling proteins. I think without, too much introduction, I think all of us have been reading Tianmin's work for the past couple of years, and his lab is really super productive. And then just, I think last week, or no, this week actually, Timmy just have a new paper online on Cell, which describing the DNA assembly in microbiome. I mean, it's really exciting work. I think we are very lucky to have Timmy with us today. And thank you so much, Timmy. And we are very much looking forward to your talk.
Thank you, Chen, for the very nice introduction and for inviting me to share our work with the community. As Chen said, a few years ago, I gave a talk mainly focused on my post research on the DAC receptor. After I started my lab, as Chen said, I focused on lysosomal signaling and also spent quite a bit of efforts on immune signaling. But today I won't talk about lysosome part, mainly talk about the immune part. So let me start by thanking all the people who worked day and night in the lab to generate all the data that I'm going to tell you today. So we have a pretty small lab over the past 3 years, but this year we expanded from 3 people to 6 people, with 2 postdoc and 4 grad students. They are really good people who have passion in science and produced amazing data. So as Tran said, we are interested in cell signaling by identifying new signaling modality and uncovering novel mechanism. We hope we have a chance to develop some new therapies in the long run. Regarding the mechanism of cell signaling, let me use this slide to tell you the classic view of cell signaling. As shown here, we all know ligand-induced conformal changes or receptors actually initiate cell signaling or signaling cascade represented by the G protein coupled receptor. This is really a class model of how cell signaling initiated. During my postdoc training with Professor Hao Wu, first at Weill Cornell and then at Harvard Medical School, using the death receptor signaling pathway, which I talked about a couple years ago, we actually highlight the functional importance of those high-order assemblies in cell signaling. Based on my research and others, we propose a new paradigm in cell signaling in which ligands, receptor, adapter, and effector proteins assemble into a supramolecular assembly or higher order assembly to initiate cell signaling. This actually contrasts the classical model I just described to you a minute ago. So compared to a classical signal model, this new mechanism actually allows cells to display cooperativity and a threshold response to ligands or stimuli. Additionally, this mechanism also allows cells to reduce or filter noise. This is quite important for immune response because we know immune response actually is important for our health. But again, over response also causes lots of trouble to us. That's why we believe immune system actually evolved this very unique mechanism to precisely control the activation of immune response. So after I started my lab, actually I switched to a complete different area from what I did as a postdoc. As Chan said, one focus on lysosome signaling, which I'm not going to talk about today. The other focus is really focused on bacterial immune response for two reasons. First, bacterial immune response actually resembles human immune response. Studying this system has a chance to provide insights from evolutionary perspective. Second, many bacterial immune system holds great potential to be reprogrammed into valuable molecular tools. One example is CRISPR-Cas9, which evolutionized the entire genome editing field. Today, I'm going to use 3 examples to tell you how our study have expanded our understanding of immune signaling. In particular, we believe some of the system actually challenge the high order assembly media signaling paradigm, which I contributed to establish as a postdoc. So the 3 systems I'm going to talk about are called Sparta, DDMDE, and the ATP containing system. Both Sparta and DDMDE are involved in antiplasmin defense. Additionally, both of them actually has Argonaute protein. In contrast, this ATP containing system are mainly involved in antiphage defense. Furthermore, the Sparta got activated through ligand-induced oligomerization, while DDMDE is activated through a dissociation mechanism. However, the ATP-containing system, they form pre-assembled oligomer for antiphage defense. So let me start with introduce you Argonaute. As many of you are familiar with Argonaute, this is a eukaryote argonaut that contains a few functional domains: end domain, path domain, mid domain, and the pivot domain. You can see here the mid domain functions to recognize 5' phosphate or guide RNA when the target RNA comes in. The pivot domain, which is a nucleus domain, can cleave the target RNA. This process is known as RNA interference. Different from eukaryote argonauts, bacteria has two different types of argonauts, long argonauts and short argonauts. The long argonauts in bacteria resembles eukaryote argonaut. In contrast, short argonauts only contains two domains, the mid domain and the pivot domain. Furthermore, the pivot domain in short argonauts has no nucleus activity. Therefore, short argonauts were believed to be non-functional for years. More recently, bioinformatics analysis showed short argonauts tends to associate with the partner proteins. For example, the system that I'm going to tell you called Sparta contains short argonauts and a partner protein called tier A path, which contains the N-terminal tier domain and the C-terminal A path domain. For those of you who are familiar with innate immunity, tier domain is a very popular adapter domain in human innate immune system. The A path domain was believed to be a homologue of the path domain in long argonauts. 2 years ago, a European group published a paper to describe the function of Sparta. As you can see here, upon recognizing a guide RNA and a target DNA, the Sparta can form an oligomer, potentially through tier-tier interaction. The oligomerized Sparta can hydrolyze NAD, eventually leads to bacterial cell death. This is a mechanism known as abortive infection, which is a very popular mechanism for anti-plasmid or anti-phage defense in bacteria. Inspired by this work, we decided to use cryo-EM single particle analysis combined with the biochemical analysis to really understand the assembly and activation of the Sparta system. In the next few minutes, I will first describe structures of aposparta, followed by Sparta in complex with RNA and the Sparta tetramer. I will conclude with the assembly and the catalytic mechanism of Sparta. So indeed, the short argonaut and its partner protein tier A path form a very stable complex. Within this complex, the A path domain form extensive interactions with both the mid domain and the pivot domain, while the tier domain also packed very tightly against the mid domain. Additionally, we identified a very unique C-terminal motif in Sparta system. This motif occupied the nucleic acid binding pocket, potentially inhibiting the nucleic acid binding. To understand the functional importance of this motif, we further determined the structures of monomeric Sparta with RNA and DNA. You can clearly see the target DNA actually can directly compete with the CTM motif. Consistently, Sparta with a truncated CTM actually develop much higher binding affinity towards DNA. Together, this data actually suggests that CTM potentially functions to block the DNA binding and inhibit the Sparta autoactivation in cells. Furthermore, The RNA DNA binding actually trigger dramatic conformational changes in the mid domain, which is critical for the oligomerization of Sparta. I will tell you in a minute. As I said, to understand how the Sparta form oligomer, we further determined the oligomerized Sparta. You can clearly see four protomers assemble as a tetramer, resemble a beautiful butterfly. In this tetramer, we actually have two different conformational state of Sparta protomer. Therefore, this is an asymmetric structure. You can clearly see Sparta protomer in state 1 and state 2 assemble form an asymmetric dimer. Then two pairs of asymmetric dimer form a tetramer through tier-tier interaction. From state 1 to state 2, we saw the tier domain rotated about 180 degrees, while other domains remain the same. The rotation of tier domain allow two pairs of tier to form an asymmetric tier dimer through a dimeric interface. Then two pairs of asymmetric tier dimer form a tetramer through a tetramerization interface. Together, the dimeric interface and the tetramerization interface contribute to the tier-tier assembly. Other than the tier-tier interactions mediated by the mid-domain is also critical for the assembly of the Sparta tetramer. As I said, RNA and DNA binding actually trigger dramatic conformational changes of the mid-domain. The consequence of the conformational change actually leads to the exposure of a very positive charged pocket. This pocket can engage with a very negative charged motif from a neighboring promoter to form a symmetric Sparta dimer. So far, I have tell you or describe structures of Sparta in apo state, a monomeric state with RNA and DNA, as well as a Sparta tetramer. So with this structure, we propose a potential mechanism of Sparta assembly. We believe Sparta in apo state is an auto-inhibit state because the CTM occupied the nucleic acid binding pocket. When DNA RNA comes in, the DNA molecule directly compete with the CTM to kick out CTM from the nucleic acid binding pocket. Meanwhile, trigger dramatic conformational changes in the mid-domain. Therefore, the mid-domain mediates the assembly of the symmetric Sparta dimer. Somehow, one tier domain in the symmetric Sparta dimer rotate about 180 degrees to form an asymmetric Sparta dimer. Eventually, two pairs of asymmetric Sparta dimer form a tetramer through tier-tier interaction. You may ask whether we could capture structures of the asymmetric and symmetric Sparta dimers. After overcoming numerous technical problems, we eventually obtained a very high resolution asymmetric dimer and a moderate resolution symmetric dimer. So with all these structure enhanced, we are confident to see that the Sparta tetramer is formed through a hierarchical assembly process, starting from auto-inhibitor Sparta monomer, all the way through monomeric RNA bound Sparta, symmetric Sparta dimer, asymmetric Sparta dimer, and a tetramer. So Next, we ask why the symmetric dimer transits into an asymmetric dimer. How sparta tetramer catalyze NAD hydrolysis. To address the first question, we analyze structures of symmetric and asymmetric sparta. You can clearly see um, in this symmetric tier dimer, the two-tier domain actually has charge repulsion. We believe this charge repulsion drive the rotation of one tier domain to rotate about 180 degrees to form a very stable asymmetric tier dimer with charge and shape complementarity. The next question is how sparta tetramer catalyze NAD hydrolysis. To address this question, we further determined the structures of sparta with NAD substrate. You can clearly see in the tetramer, we have two catalytic centers. The NAD molecule are tightly associated on the interface of the asymmetric tier dimer. The question we ask whether this asymmetric sparta dimer is necessary and sufficient for catalysis. To address this question, we introduce a mutation to the tetramerization interface. Therefore, we can purify very stable sparta dimers by biochemical assay. You can clearly see those mutant or dimeric mutant has no activity at all compared to the wild type. So these experiments really highlight the functional importance of tetramerization in catalysis. So with that, let me summarize what I just taught you about the Sparta story. So as I said, the Sparta form a tetramer for catalysis through a hierarchical assembly process, all the way from autoinhibitor monomer through substrate bound or arginine bound monomer, symmetric dimer, asymmetric dimer, eventually form a functional or catalytic active tetramer. So now let me switch gears to tell you another story, which is also Argonaut containing anti-plasmid defense system called DDMDE. So this is a very novel anti-plasmid defense system identified in a pandemic Vibrio strain 2 years ago. The genetic analysis showed both DDMD and DDME are required for anti-plasmid defense. So when I saw the paper, I made a structure prediction, used the AlphaFold, which was thought will take over our jobs in a few years. You can clearly see, so DDMD contains two domains, a nucleus domain and a helix domain, while the DDME adopts a typical Argonaut fold. DDME contains the N domain, Gas domain, mid domain, and the pivot domain. Notably, there's an inserted domain in the pivot domain. We later on coined this domain as DID, which stands for DDMD interacting domain. As I said, the genetic analysis showed both D and E are required for anti-plasmid defense. The first question we ask whether these two proteins can form a stable complex for anti-plasmid defense. You can clearly see we are so disappointed that the two proteins failed to form a stable complex on gel filtration. As I said, DDME adopt Argonaut fold. We all know Argonaut like nucleic acid. Therefore, I hypothesized the two proteins myoformal complex in the presence of nucleic acid substrates. Therefore, we screened numerous nucleic acid oligos, and eventually we were lucky enough to obtain a stable complex of DDMD with the magic DNA substrates. I still vividly remember the excitement we had when we saw the co-migration of the ternary complex on gel filtration. We then went on to determine the cryo-EM structure of this ternary complex. You can clearly see within this complex, one copy of DDME was linked together with a dimeric DDMD, or two copies of DDMD. So now let's look at the structure together. Within this structure, we actually saw 3 DNA fragments. Based on our knowledge of Argonaut, we named the 3 fragments as guide DNA in green and complementary target DNA, or ctDNA, in cyan. And this yellow one, or orange one, we named as non-complementary target DNA, or nctDNA. You can clearly see the DDME binds to guide DNA, ctDNA, nctDNA, and making a bubble. So the single-strand part of this nctDNA extended out to the DDMD. Therefore, this magic substrate had a chance to link the DDMD and the DDME together. Furthermore, this structure immediately suggests that DDME is a DNA-guided DNA targeting modular. So to test this hypothesis derived from structural analysis, we did those EMSA assay. You can clearly see DDMD, a DDME actually prefers a 5' phosphated guide DNA to target a DNA molecule. Guide RNA or targeted DNA cannot be effectively associated with the DDME. Furthermore, we use EMSA assay to show DDME actually prefers a guide DNA shorter than 14 nucleotide. This actually is also consistent with our structure analysis, because a longer guide DNA will form a structure clash with another functional domain of DDME. Additionally, through structure analysis, we realize there are 2 catalytic aspartate like in TT-Argo was replaced with serine or methionine in DDME, indicating DDME is catalytically inactive. Further biochemical assay actually confirmed DDME indeed is an inactive nucleus. Structural analysis further showed that DDME actually used the very unique DID domain to recruit DDMD, mainly through charge-charge interactions. To understand the functional importance of this interaction, we developed both in vitro and in vivo assays. We first used in vitro plasmid elimination assay to show both DDMD and DDME are required for plasmid elimination in the presence of a guide DNA. Furthermore, we developed in vitro assay. You can clearly see we put DDMD in a kanamycin-resistant plasmid and DDME in a ampicillin-resistant plasmid, then co-transfect the plasmid into E. coli. Upon IPT gene induction, the two proteins will be expressed in E. coli to eliminate plasmid. Therefore, you cannot get any colony on place with antibiotics. Using this in vivo assay, we showed we first confirmed that both DDMD and DDME are required for anti-plasmid defense. Furthermore, if we mutate those residue on the interface of DDMD and DDME, we can clearly see all those mutants significantly reduced the anti-plasmid defense activity of the system, highlighting the functional importance of DDMD interaction in plasmid elimination. So the last question we ask is how DDMD get activated in the system? To address this question, we first determined the structures of ApoDDMD. We found that the ApoDDMD assembles as a dimer. Furthermore, as I said, DDMD actually has a helix domain. All his homologue actually assemble as a monomer. DDMD adopt a dimer with the DNA exit site right located at the interface. Therefore, this assembly actually block the DNA exit pathway, indicating ApoDDMD actually resemble an autoinhibitor dimer. Additionally, we noticed the DDMD form a dimer through two interfaces we named as interface 1 and interface 2. If we compare structures of ApoDDMD and DDMD in complex with single-strand DNA, we can clearly see DDMD with DNA substrate actually significantly expanded. This expansion actually reduced the interaction interfaces, also disrupt many interacting pairs at the interfaces. This suggests the DNA binding potentially trigger DDMD dissociation. To test this hypothesis, we developed EMSA assay again. Using this assay, we found that a forked DNA substrate with a 3' overhang or with a long 3' overhang can effectively trigger the dissociation of DDMD dimers into monomers. Furthermore, we did extensive structural analysis to identify the partially dissociated DDMD and fully dissociated DDMD upon DNA substrate binding. You can clearly see in this partially dissociated complex, the interface 1 is still preserved, while interface 2 is completely gone. So this data actually suggests the DDMD dissociation may undergo a two-step process with the interface 2 being disrupted first, followed by interface 1. Therefore, we speculate DDMD dissociation via two-step sequential mechanism eventually leads to the formation of DDMD monomer. So this monomer has a chance to have higher activity or helix activity, allow DDMD to effectively translocate along plasmid for cleavage. So in the interest of time, I cannot tell you all the other data we produced in the paper, but let me use these summary slides to tell you what we found. As I said, the ApoDDMD represent an autoinhibitor state with the helix domain, with helix activity being inhibited. However, actually, ApoDDMD still have residual nucleus activity, although not very high. This nuclear activity allows DDMD to convert a supercoiled plasmid into a nicked circular plasmid, facilitating the targeting of DDME. DDME require a short guide DNA to target this plasmid. Upon engagement, the DDME make a bubble on the plasmid for the recruitment of DDMD. DDMD was recruited as a dimer onto plasmid. Then the DNA binding can effectively trigger the dissociation of DDMD dimer into monomers. The DDMD monomer can effectively translocate along plasmid to cleave the plasmid into small fragments. We believe these small fragments will function as a guide DNA to further serve as a positive feedback loop to enhance, to further trigger the activation of the system and eventually allow the system to completely eliminate all the plasmids in a cell or in a bacteria. So I will use the last few minutes to summarize the ATP-containing system we studied in anti-phage defense. So as you can see here, this system was identified through bioinformatics analysis a few years ago when I just started my lab. When I saw this system, I realized all the system contains two components. One component is ATPs, and the other component is an enzyme with diverse enzymatic activity. This immediately allowed me to link the system with the human inflammasome. For those of you who are familiar with the inflammasome, inflammasome oftentimes they contains the ATPase domain that allow the inflammasome to form oligomer. The assembly of the inflammasome can effectively trigger the activation of caspase-1. So therefore, I would hypothesize in bacteria, this ATPase containing system may use the ATPase component as a sensor to trigger the activation of this enzymatic partner for anti-phage defense. As I mentioned, those inflammasome actually use ATPase domain to form those ring-like oligomer. This oligomerization will further trigger the oligomerization of the CAR domain for recruitment of downstream caspase-1 through card-card interaction, eventually leads to the activation of caspase-1 for processing interleukin or gastrin D to trigger immune response. We hypothesize those ATPase containing system in bacteria may also use the ATPase components to form an oligomer, which will serve as an effective platform for the activation of the enzymatic partners. Indeed, our structural analysis showed so all those components actually indeed use the ATPase components to form an oligomer. And also, very unexpected discovery is that we found the two components actually form a pre-assembled oligomer, contrasting the inflammasome. For those of you who are familiar with the inflammasome, you may know actually inflammasome only in the presence of pathogen, some part of the pathogen or molecular patterns actually can trigger the oligomerization of the inflammasome. Then the inflammasome can further recruit downstream caspase to activate caspase. This bacterial system is quite different. So both components actually form a pre-assembled oligomer. But all these oligomers stay in an inactive state. They need the phage components to trigger their activation. At the moment, we still don't know which part of the phage can switch these pre-assembled oligomer from an inactive state to an active state. You can clearly see those systems, they have lots of structural similarity with the inflammasome. But again, their assembly is, their overall architecture is quite diverse compared to the ring-like oligomer inflammasomes. More important, as I mentioned, this system form a pre-assembled oligomer instead of a ligand-induced oligomer in inflammasome. I believe all those studies has significantly expand our understanding of immune defense in bacteria. So with that, let me summarize. So today I actually use three systems to tell you bacteria actually evolve much more complete mechanism to drive immune defense. With that, I want to thank my trainee again, and as well as my fantastic collaborator, funding agency, as well as numerous cryo-EM center allow us to collect data to uncover all those unexpected mechanisms we never thought about before. So I think it's about time. With that, I thank you for your attention. I'm happy to take questions you may have.